Hey, Mr. Lindbergh. My name is Liam Poole. I am alone in this project, and this is my video on mitosis. So, we of course first must begin an interphase before mitosis starts. This is essentially the G1 phase, where we have our cell, the cell membrane, our nucleolus, the nuclear envelope with the nuclear membrane, and our mass of chromatin, which is the nucleolus, really. And this chromatin, once it enters the S phase, starts to duplicate, and it's starting to form these long chromosomes by slightly condensing. And eventually this nucleus will turn something like this. Then we move into mitosis, where we begin with prophase. And prophase is the condensing of the long strand chromosomes into sister chromatid pairs, which I will demonstrate by cutting these up. Now that we have our sister chromatids all assembled through the condensation of the chromosomes, and the nuclear envelope has been kind of gone away with because all of the chromosomes have condensed and we have all the DNA and sister chromatid pairs, our mitocentrioles are at the poles of the cell, and we can now enter the second phase, metaphase. This is where the sister chromatid pairs line up across the middle of the cell. And forgive my poor model, they might have to fit a little outside of the reaches of the cell into desk cell parts. And what the microtubules will do is attach at the centromeres. All right, now, so our microtubules have successfully attached to the centromeres of our chromosome sister chromatid pairs that are lined across the middle of the cell. Now, we will enter anaphase. Anaphase is where the microtubules, now that the chromatid pairs are on the right side, essentially, are able to pull apart and separate the chromosomes onto either side. So separate the chromatid pairs at the centromere so that they can become their own chromosomes. Been completed, as these have been drawn apart, we enter the final stage of mitosis, which is telophase. In that telophase, we see the chromosomes attached to the microtubules draw closer and closer to the centrioles. And in this MTOC kind of area, the poles of the cell, and this bringing together all of them starts to reform the nuclear membrane. So that comes into play here. And at the very same time, the chromosomes start to decondense back into loose chromatin, which I will demonstrate by cutting the chromosomes back up and putting them into this nuclear membrane. We also start to see now how the cell membrane begins to pinch and get just a little bit closer to separating the two. Cytokinesis! And so in cytokinesis, we see a cleavage furrow begin to form, made by the cell, which is almost like perforations, priming the cell to be fully separated and divided by its two nuclei. And then, of course, cytokinesis. And this is in an animal cell, to be specific. And at this point, the process of cell division is complete. In a plant cell, cytokinesis differs as a plant cell is not as malleable because of its hard and rigid cell wall, as shown by these hard, sharp edges. And so in order to conduct cytokinesis, what happens is that small vesicles start to form across the center of the cell, separating the two newly formed nuclei. And this forms what is called a cell plate. And the cell plate is a way for the cell to also conduct cytokinesis. Because with the cell plate, it's there, the vesicles are there, and it's able to separate just like a cleavage furrow would. And so now you have two totally independent diploid plant cells with hard and rigid cell walls on both of them. This has been me and Poole. Thanks for watching. Goodbye.